Right, welcome back. Here's our second AS level recapping lesson on another group of very important biological molecules, the nucleic acids. But we'll also have a look, besides the nucleic acids, we'll look at one of those nucleotide derivatives, ATP. We'll also look at water and briefly mineral ions as well and their role in biology. Okay, but we're going to start off looking specifically at our nucleic acids. So nucleic acids are another group of biological molecules. They're polymers and their monomers are called nucleotides. Okay, so I'm going to draw, to start off with, a basic nucleotide to give you an idea of what their general structure is. You'd have to be able to do this yourselves in the exam, so you've got to draw the symbols and the accurate kind of positions of where the bonds would occur. So I'll start off with, I'll write a little title of nucleotides, okay, and then I'm going to draw my phosphate group there, okay, that'll come off with a bond on it there, and that will attach to a pentose sugar of some description. So you've got to draw a pentagon like that and make sure that the phosphate group is bonded to that part of the pentose sugar, okay. And then the final component of our nucleotides is our nitrogenous base there. So that's our general structure of a nucleotide. There are 5 different versions of nucleotides. All 5 versions will have a phosphate and a pentose sugar of some description. But where they will vary will depend on what that nitrogenous base actually is. They could also potentially vary in terms of what sugar this is. So I'll start by talking about the 5 different nitrogenous bases. The first one is adenine. The second one is thymine, the third one is cytosine, the fourth one is guanine, and the fifth one is uracil. Okay, so they're the 5 nitrogenous bases that could be, one of these could be on this nucleotide. Okay, nucleotides are the monomers, the building blocks that make up the nucleic acids, the more complex polymer versions of these substances. There are 2 main types of nucleic acid. There is DNA or deoxyribonucleic acids, or there's RNA, which is ribonucleic acids. Okay, so they both are made up of nucleotides, but where do those DNA and RNA nucleotides differ? Well, they differ primarily on that pentose sugar. In DNA, that pentose sugar is called deoxyribose, and in RNA, it's just simply called ribose, and that's what gives them their names. Okay, so bear that in mind. Now in the exam, if they ask you about describing the structure or labelling a diagram such as this, or what is this part, never ever write pentose sugar. That's not sufficient. Be specific. Either write deoxyribose if it's a DNA nucleotide, or ribose if it's an RNA nucleotide. That's very, very important that you do that. Okay, so that's a nice general overview of what our nucleotide structure is like. Now we're going to start talking about how I can start to assemble these into those nucleic acids. So now we're going to look at a little bit more detail of those nucleic acids and actually how we can assemble those nucleotides into the polymer nucleic acid. So what I've drawn here are 2 separate individual nucleotides. Okay, in order for them to bond, a condensation reaction has to take place, and a bond will occur between the phosphate group of the first nucleotide and the ribose sugar. In this case, let's assume it's deoxyribose, we're talking about DNA, and another bond will form there. So that bond there is called a phosphodiester bond. So to recap, actually, we've come across quite a few bonds now in our biological molecules. We've seen glycosidic bonds in carbohydrates, we've seen ester bonds in lipids, we've seen peptide bonds between amino acids, we've also seen hydrogen bonding in our proteins and disulfide bonds and ionic bonds, and here's the one for our nucleic acids. We call it a phosphodiester bond, and this will obviously continue over and over and over again, potentially. If you think about it like this, you know, your DNA is up to 2 meters long inside each of your nuclei of your cells as you unwound it and straighten it out in a linear fashion. So these DNA is an incredibly long molecule, but I'll talk a bit more about the structure and properties of DNA a bit later in the video. So bear that in mind, a nice little phosphodiester bond there. So I'm just going to rub this one out and move on to looking at different types of RNA. Okay. So RNA, there are actually 3 different types of RNA. The one you'll probably most come across, more often than not, is mRNA, but there also is tRNA and there is rRNA, just to make things confusing. Always the first letter of each of those is lowercase, followed by the capitalized RNA. So mRNA's role is to effectively form a message, a copy of a section of our DNA, basically a copy of a gene. That can then be used to create a protein based on that DNA, that base sequence from our DNA. tRNA, there are several types of tRNA. Each one will have specific codons and anticodons attached to it, and its job is basically to shuttle over a specific amino acids that are going to correspond to that mRNA sequence, and that way the primary structure, that specific sequence of amino acids in our protein, can be assembled. Our RNA is used to actually help form the ribosomes themselves, those organelles that are there for protein synthesis. So actually I think of RNA as almost kind of bridging the gap between DNA and proteins. It's all about kind of the machinery that helps to transcribe and translate DNA-based sequences into polypeptide sequences. Okay, so that's a nice little simple overview of RNA. Now let's look at DNA in a little bit more detail. Before we kind of delve deeply into it, I think it's quite important that we acknowledge those people, those very important scientists who actually played a role in determining the structure and, you know, the actual importance of DNA. Those are Watson and Crick, but also Rosalind Franklin as well, and her work with her X-rays and so forth really kind of help us understand the structure of this molecule and the important part it played in biology. Now I could wax lyrical for a very, very long time about how enamoured I am with DNA as a molecule. It really is quite extraordinary, and it is really the essence of life itself. It's the only known molecule in the universe that we know of at this point that is able to self-replicate. Not only is it able to copy itself, it also does so incredibly accurately, and that's an important word when you're talking about DNA replication. It does so accurately. Occasionally errors do creep in, which we call mutations, but the structure and the mechanisms that surround DNA really do keep those to an absolute bare minimum. So it really, really is an extraordinary molecule. Without DNA, there is no life. It is the essence of life. And fundamentally, that's what natural selection is acting upon, is our DNA sequences, because they underpin everything. So let's start looking at the structure of DNA. So DNA, it's described as a double helix. It's 2 basically spirals wound around each other. The backbone is what we call the phosphodiester backbone. So when I showed you the nucleotides before, that's where your alternating deoxyribose and phosphate deoxyribose, phosphate makes up that backbone. And obviously between each of those nucleotides is an ester bond. So this outer double helix actually has, the main role really is to actually create and improve the stability of the molecule. All those many bonds are very, very strong, so it's very difficult to break and fragment the DNA strand, so it remains intact. By coiling in the way it does, it also protects those potentially vulnerable nucleotides from any kind of chemical or physical corruption. So that aids in the stability and also maintaining that key genetic information, because that's what DNA is for. It's the genetic information molecule for life, effectively. So very, very, very important indeed. What actually holds the 2 DNA strands together, keeping this double helix? Well, it's actually the nitrogenous bases. And the nitrogenous bases form what we call complementary base pairs. And they do so in a very, very specific way. Adenine will always form a complementary base pair with thymine, and cytosine and guanine will always form a complementary base pair as well. What actually forms that base pair is actually hydrogen bonding there. So I've drawn those dashed lines to represent that. Adenine always bonds with thymine, and cytosine and guanine. This is always the case in DNA. I could look at any single living organism on the planet, and the genetic code is universal. All living things, no matter what they are, use this system. Okay, so it's quite remarkable. There is no other software version, if you like, for life on this planet. It's all this way, and there's no other way of doing it. When you're looking at RNA, the base pairing rules are a little bit different, because instead of thymine, we've got uracil instead. So we haven't got that nitrogenous base present. So in fact, when, say, mRNA is forming and making a complementary copy of the DNA base sequence, if you had adenine on the DNA as one of its bases sticking out, uracil would form a bond with it. Oops, I can't spell. I'll put C there. Would do that like that. Cytosine and guanine, the same in DNA and in RNA, so that would be the same. So that's the only kind of major difference there between DNA and RNA in terms of the base sequencing. So why is complementary base sequencing important? It's one of the most important parts of the structure and therefore the properties of DNA. Well, what it allows, first of all, is to form many hydrogen bonds, billions potentially, and that obviously increases stability of the molecule. Many hydrogen bonds together are very strong, so it prevents the two strands separating. But it does allow very accurate replication, semi-conservative replication, which we'll talk about later in the video. So it's important to word it that way, accurate replication. It also can hold genetic information. It allows transcription to take place, i.e. the conversion of the DNA's base sequence into an mRNA sequence, therefore it can be used to translate into protein. So that base sequencing is very important indeed. Okay, so there's a nice kind of basic overview of the structure and the properties of DNA as a molecule. We're now going to look at this idea of semi-conservative replication. How is this molecule able to replicate itself accurately and efficiently? Cheeky tea break. Delicious. Beautiful that. Right, okay then. DNA replication, more specifically semi-conservative replication. So, to start things off, there are two very important enzymes that are involved in DNA replication. The first one is called DNA helicase, and what DNA helicase does, it will break the hydrogen bonds between those complementary base pairs. Now, individually, they're quite easy to overcome. So, almost think about unzipping. It's kind of what's going on there. So, helicase would have come through and start to unzip the two strands, separating them out into two template strands. The next important enzyme is called DNA polymerase. But I'm not going to talk about DNA polymerase yet because we're not quite at the right stage. So we'll take it step by step. So step one, the DNA strand would start to uncoil. Helicase would break the hydrogen bonds between them, separating it into two template strands. Always write that, two template strands. Now these strands are open, and they will allow free nucleotides that would be loose there within the nucleus to form complementary base pairs. So you would find that an adenine would form a hydrogen bond there with a complementary base strand. We would call this the template strand here. And what I'm forming here, well, eventually I'll end up forming is the replica strand. So I'd have a T there, a G there, and so on. We haven't obviously unzipped all the way up here yet, but we will do eventually. Okay, like that. So that's all my free nucleotides. They're loosely in place at this stage. There's only hydrogen bonds holding them in there, but they have formed their complementary base pairs. I'll just sort of demonstrate that like that. What actually will form the new phosphodiester backbone on our replica strand is that second enzyme, that DNA polymerase. And what DNA polymerase will do, it will bind to a hydroxyl group on carbon 3. It will bond there, and it will then join together those two adjacent nucleotides, forming a phosphodiester backbone. So if I do it like that, and imagine that like that. So what would eventually happen I'm not going to draw all the way up here, but you can appreciate that that strand would carry on unzipping because of helicase. More free nucleotides would come in, form them a complementary base pairs. DNA polymerase would come along and join together and form phosphodiester bonds between all those adjacent base pairs. And what we'd end up with is two separate DNA strands from the original. Now, the reason we call this complementary base pairing is because we still have one half of the original DNA strand within the new DNA molecule, and half of it is a new strand. So we've got this template strand and a replica strand, if you like, bonded together. That's one molecule. And then the new one, the second one, sorry, will have half template, half replica strand. And there have been various experiments done, particularly using isotopes of nitrogen that can be used to identify this is actually the way that DNA replicates. I'm not, for brevity's sake, I'm not going to get involved in those in this video. Feel free to have a look at those. They are very important. They come up a lot in the exams. But I'd like to move on now from our nucleotides to a nucleotide derivative, and that is ATP. So I've mentioned that ATP was a nucleotide derivative. It doesn't form polymers, nucleic acids like our RNA or a DNA, but it is very similar in the structure to our general nucleotides, but it has a very, very different function. So let's look at the structure of it first of all. First thing to note is it has got a nitrogenous base. Specifically in this case, that is adenine, always adenine, hence the name ATP, which stands for adenosine triphosphate. It has a pentose sugar, which is always ribose. And instead of having just one phosphate group, it has three of them. So all together, it makes a structure. So adenosine triphosphate. The adenosine is referring to the adenine and the ribose, and then the triphosphate is there. So ATP is an incredibly important molecule inside of our cells. It is the it's a universal energy currency of cells. All living things, from the tiniest bacterium to the mightiest melt like me, within our cells requires ATP as an energy source. Now, why don't we just use glucose directly? Well, glucose is great. It's got lots of potential energy in it, and it's our primary energy molecule. But in order to get the energy out of glucose, it requires a lot of complex enzyme-driven metabolic pathways, a lot of steps basically to get the energy out. Whereas ATP can provide an immediate energy source. Okay, it's a small molecule, it's soluble, it can move within the cell very easily, and it can release immediate amounts of small, controlled amounts of energy. In addition to releasing energy from this like high energy bond here, it can also use that phosphate and actually kind of activate molecules. So it can actually activate them to make them more likely to react in future reactions. And when we get into things like glycolysis in respiration in year 13, you'll see a specific example there of where ATP is used to activate a molecule. But I'm not going to go into much further than that. Another couple of key points to look at are, how does ATP form? So to form ATP, we have to start off with ADP, which stands for adenosine diphosphate. So it's missing one of the phosphates that is required to form ATP. It will then bond with another phosphate group here, which I've demonstrated with this sort of general formula we use for phosphate in these reactions. And the forward reaction is to form that ATP. But you'll notice that this is a reversible reaction. So ATP can be hydrolyzed and then rephosphorylated over and over again between, hydrolysis and phosphorylation, over and over and over again, which is great, which means that that's another key feature of ATP, is that it can be regenerated and recycled. You may only have a few grams of this within your body at any one time, but throughout the day, you're probably going to make, somebody like me, maybe 40 or even 50 kilos of the stuff. Really, really quite awesome, I think, that fact there. So again, as its role as that immediate universal energy currency molecule. Okay, so that's ATP. We are now going to look at water, its properties, and how its structure relates to its properties, and why that is essential for all life on Earth. Okay. Right then, water. So hopefully you're familiar with water. It's chemistry a little bit, H2O. And I always think it looks a bit like Mickey Mouse. Hopefully I won't get flagged by Disney by mentioning his name, but there we go. And what's special about water? Well, it's a simple covalent molecule like carbon dioxide, ammonia, hydrogen, nitrogen, whatever. But it is quite unique in them. It's the only, at least the only chemical that I know of on Earth that can exist in all three states at any one time on Earth. So you've got solid water, you've got liquid water, and you've got gaseous water, all existing on Earth at any one time. I don't believe any other substance on Earth exists in all three states naturally. And a lot of that is down to the structure of water. It's what makes it kind of a little bit different than its cousins, the other simple covalent molecules. And that is because it's a polar molecule. So it has a difference in charge from one end of the molecule to the other. So the hydrogens are referred to as delta positive. Now I could delve deep into the chemistry here. I'm not a chemist, I'm a biologist, so I'm not going to go too much there. But it's all to do with the distribution of the electrons. Here we've got the proton that was in the hydrogen atom is closer to the outside, so it gives it that overall positive charge. Whereas the oxygen end of these molecules is delta negative, so hence that polar name, because we've got positive and negative ends on them. And what that does is it creates an attraction between the positive and the negative, and we actually form hydrogen bonds like that through that attraction. And that effectively kind of makes water sticky on the molecular level. It's cohesive, it wants to stick together, it wants to stay together, and that gives water a lot of really unique properties, many of which are essential for life on Earth. If water behaved in a different way down to it didn't have its bonding, life itself would not be possible. I've mentioned many times that carbon is the element of life and DNA is essential for life, but water really is too. Without liquid water, you cannot have life. So it is really, really essential. So what does this hydrogen bonding and this polarity actually lend itself to biology? Well, the first thing to look at is the idea of specific heat capacity. So specific heat capacity is a measure of how much energy is required to heat up 1 kilogram of a substance by 1 degree C. So metals have a low specific heat capacity, so they don't need a lot of energy to heat them up, to raise their internal temperature. Whereas water actually has a very, very high specific heat capacity. Off the top of my head, I think it's something like 4,200 joules per kilo degree C, whereas something like copper is only like 300 joules per kilo degree C. Quite complex unit specific heat capacity. Now, what does that mean? Well, in other words, it takes a lot of energy to raise 1 kilogram of water by 1 degree Celsius. So that means it's basically kind of much more thermostable in that respect. Now, if you live in an aquatic environment, that's really important, because if you lived in something like a pond, for example, imagine a little tadpole swimming along a lovely little pond. If water had a low specific heat capacity, during the day, when the sun came out, the water would heat up really quickly, potentially denaturing the enzymes in the tadpole and killing it. Likewise, at night, as the temperatures dropped, the water would lose that energy very quickly as well, and it would potentially freeze, become a solid very, very easily. But because water's got a very high specific heat capacity, it means its temperature doesn't fluctuate very much, it keeps very stable. And then we expand that to a large body of water, like the world's oceans, their temperatures are really, really stable, really, really ideal for keeping temperatures buffered. This hydrogen bonding, like I've mentioned before, makes water cohesive. It wants to stick together. It coheres and adheres to surfaces as well. So that's really important for plants, for when they're doing things like transpiration. The water molecules will cohere to each other and adhere to the xylem vessels themselves, and are able to be pulled up through the xylem, so enabling the plant to transport water up to 100 meters tall in something like a coast redwood, and that is purely down to the properties of water. If it didn't do that, that would not be possible. Also linking to specific heat capacity is the latent heat capacity of water as well. So water requires a lot of energy for it to evaporate, to boil off. So it wants to basically stay as a liquid, which is ideal as a medium for life, because water is an amazing solvent. It enables many substances like mineral ions, etc., glucose, to dissolve in it, which means they are free to move around the cells. That obviously aids in various chemical reactions. Water itself is somewhat reactive as well, and we've seen in condensation and hydrolysis reactions, when we looked at biological molecules, the role that water played there in terms of as a chemical reagent. So water itself is, really important for life. Without it, wouldn't be possible. I've suggested that potentially, if you wanted to, you could look at how ice forms and how that is also essential for life. Now, that's certainly beyond the specification, but it's something you could throw into the essay there if it was appropriate and relevant to the topic. And the way that ice forms is pretty, it's fairly unique, certainly that I know of in simple molecules. And again, that also helps with life as well. If ice formed in a different way, we would be in real big troubles. Actually, our oceans would freeze from the bottom up, which would be catastrophic, but instead it's the surface that freezes, and that's obviously to do with the density of solid water compared to liquid water. But I'll allow you guys to look at that for yourselves. The last little bit I'm going to keep quite brief. It's something that kind of we touch on throughout the 2 years, and that is mineral ions. Okay then, so mineral ions then. Like I alluded to, these kind of crop up throughout the kind of 2-year A-level biology course, and they play a wide range of roles here. I'm not going to go into epic detail for each one. That'll be appropriate for each given topic. But something to bear in mind is always keep an eye on these and add them to your repository. Always mention mineral ions again, or whoever's teaching you, whatever crops up in your head, because they can always be used in the essay. Sometimes I've actually seen essay questions that are completely surrounding this idea of the importance of mineral ions in biology. So it's a good one to have in your repository. So some of the sort of mineral ions that are important, iron, heme, because that's obviously involved in hemoglobin, and that obviously enables it to transport oxygen in red blood cells. So iron's an important ion as well. You'll also come across calcium ions, more in year 13, and they play an important role in muscle contraction. We've got sodium ions, which we've already looked at this year when we looked at the idea of co-transport, along with potassium ions as well. So they're very, very important for the co-transport of glucose into our epithelial cells in our small intestine. You'll also see they play a very, very key role in nerve impulses, action potentials, and so forth there. So really important. We've also looked at the role of phosphate in today's lesson. So phosphate is used to help form DNA, RNA, nucleotides, so that's ATP. So you can see that phosphate again is a very, very important mineral ion. We also look at other ions in year 13 in the nitrogen cycle, like ammonium and things like that. So there's a whole wide range of them. Hydrogen ions as well, forgot to mention those. They're very, very, very important. And when we move on to photosynthesis and respiration, they're going to come up a heck of a lot. So you can see that actually with ions, you can touch on nearly every single topic within the biology course. So bear in mind their importance there. You're very unlikely to get an entire question related around them, unless it's an essay question, but they are going to crop up here and there. Okay, so that'll bring to a close our nucleic acids topic 2, all done there. So Dr. P signing off. You guys stay safe, stay indoors, get some work done, enjoy the sunshine. Happy days.